We're going to get an overview of the ethical issues of germline modification in animals from Alison Van Eenennaam, who's at UC Davis, and very, very distinguished researcher, studies animal genomics and biotechnology at UC Davis, and she basically is, I would say, one of the leading authorities in the world on animal science and gene editing. And I can tell you, as a member of the organizing committee, she did her best to keep us on the straight and narrow in terms of keeping the issues clean about what are we talking about when we talk about lab use, what do we talk when we talk about environmental release, and what are we talking about when we talk about eating gene-edited animals. So I've learned already a great deal from her. So Alison.
Thanks, Arthur, and thanks for inviting me, Kent. I think I have you to blame for this, because I'm actually an animal geneticist and a cattle breeder, and so I'm a bit of an oddball in this particular audience, and a little like Peter yesterday, I'm not an ethicist. However, I'm going to talk about the ethical issues associated with germline modification of animals, and that was the title that I was given, and I pushed back a little bit on the organizing committee because I said, well, hang on, your conference is about gene editing to modify animal genomes for research, and gene editing isn't in my title, and for research isn't in my title either. And I think my take-home slide is this one here, and I really want to talk about the fact that this particular conference is talking about one particular breeding method being used in animals for research. But there's a plethora of different purposes that one can use animals for, and they all have their own implicit ethical issues associated with them. Whether we're talking about using animals for food production or pets or producing biomedical products, there's a range of ethical issues associated with that. And I guess as a reductionist biologist, I have to understand what is it that's unique about the intersect of gene editing, animals, and the purpose that they're being ascribed to, because that really is what's unique about this editing method. And I'm very reticent to lay all of the issues associated with animal agriculture or animal research at the foot of this nascent technology, because I think there are unique intersects there, but we have to understand that a lot of the issues we're talking about and that were introduced previously are associated with the current breeding methods and the current purposes that we use animals for. And I guess to your point regarding maybe gene editing service animals to be, I don't know, better service dogs or smarter service dogs, that's what breeders do right now. We work with the guide dogs of America to breed better service dogs. We work to make all sorts of animals that are better suited to their agricultural purposes or maybe their different purposes. And so I think I'm going to really try to tease out what it is that's unique about the intersect of gene editing, animals, and research, because that to me is where the focus of this particular ethical session should lie, and try to do that in a way that makes sense. So I'm going to talk about the use of animals for research. And so being a scientist, what I did and went and looked at was what's the social science literature around the use of animals in research. And there were some good papers, I think, that kind of looked at some of the different aspects that were touched on earlier, and that is that people's attitudes towards the use of animals for experimentation is really dependent upon what they're being used for and who's the beneficiary. Is there a compelling reason to do animal research? The purpose, that is it justifiable? Is it lipstick testing or is it cancer cures? Yeah, very different ethics there. And the necessity of the research. Can you do it in a way that doesn't involve animals in some way? They generally are more supportive of using the lowest possible animal on the evolutionary ladder, if you will, to do the research that's suitable for that research. And we heard many examples yesterday where, although it would be nice to use a zebrafish, sometimes you do need to use higher order animals to get the actual phenotype that you're wanting to test on. However, the public concerns around using animals in research tends to be exacerbated when you discuss genetically modified animals. That's just a kind of a statement that's true. Once you bring in genetic modification, that kind of raises people's concern. And there's a concern about the fact that we can now use genetic modifications, however we achieve them, to actually replicate disease and potentially use more animals. And this creation of GM animals specifically for such purposes may challenge the 3 Rs that we discussed yesterday: replacement, reduction, and refinement. There's an interesting paper that came out just this year, actually, that looked at the factors that affect people's acceptance of animals in research. And generally, in this particular case, they were looking at research on pigs for agricultural purposes. There was generally support for non-invasive research, irrespective of whether there was actually any regulation associated with that. And of course, the IACUCs, any animal research you do, it has to go through IACUCs, so there is actually that regulatory framework in place, even if it is non-invasive research. But people are generally less acceptable if it is invasive, but the acceptance is increased if the animals don't suffer unnecessarily. And importantly, if the benefits outweigh the harms, and there's this kind of weighting of whether or not you should do the research based on the benefits. So there's a cost-benefit analysis, for want of a better economic term. If the research is regulated, then there's more acceptance. And if there's some type of refined use so that there's minimizing pain and suffering when there is such thing involved in the research, then that increased acceptance. But there was a low level of support for research using GM animals, so using genetically modified animals, even if there was regulation around the genetic modification. And what was really interesting, I thought, in that paper was that when people were weighing the invasiveness of research, they did kind of a cost-benefit analysis. Yes, it's invasive, but it has this benefit, so I'm doing a weighting. Whereas only a quarter of them did an analogous weighting when discussing GM animals. There was more of an outright ick rather than a cost-benefit weighting, which I thought was kind of an interesting observation. And then, I guess getting more to the point of this meeting is there a different acceptance depending upon the breeding method that was used to make the genetically modified animal? And there is actually a study where they did that, and they looked at people's acceptance of animals in research, looking at species, so are they more accepting of zebrafish or mice? And at breeding method, are they more accepting of using genetic engineering versus ENU mutagenesis? And I think the interesting observation in this study was that in general, people were very non-supportive of the use of ENU mutagenesis, which I don't know if the zebrafish community likes to hear that, but that's true because it's perceived as somehow being painful to the animal. And in fact, people preferred genetically modified animals to mutagenizing with ENU mutagenesis. And so there is some differential there in terms of what breeding method is used to produce the particular genetic defect. So I'm going to take the liberty of going through some of my subsequent speakers' animals to place them on this image that I showed earlier. And I'm going to start with Alan's beautiful glowfish here and just kind of emphasize the point that here we're talking about an aquarium fish produced using genetic engineering as a pet, right? So not research, not gene editing at this stage, but we're talking about that. So what are the kind of the unique issues associated with that particular species, that particular breeding method, and this particular purpose? And I have these kind of three intersecting of circles here because there's the overarching question of, well, should we even keep pets? Is it cruel to keep zebrafish in a tank? Should we breed animals to be pets? Is it fair to have a chihuahua? I don't know. I don't have one. And, does this trait impact the fish in some way? Are the zebrafish, I don't know, hurt or disadvantaged by glowing when you put them under black light? There's those overarching questions. And the species. So does this particular genetic engineering method hurt the fish in some way? Is it a food species? Because, of course, that has regulatory implications in terms of whether that's a safe protein. Do they pose any type of environmental hazards? And these were some of the questions that were asked when going through the regulatory process and resulting in the overall decision to have enforcement discretion regarding this particular fish. And then the breeding method. Does this breeding method pose new risks? And how else can you achieve that same phenotype in fish? And of course, you could inject them with dye, which comes with its own ethical issues. And so I guess for me, what I'm interested in is kind of this intersect here. So what is it that's unique about the breeding method, purpose, and species that we're interested in? Let's move on to Ron's fish. So this is a salmon that's been genetically engineered for food purposes, right? Not research. So what are the questions that come into play there? Well, should we eat fish or animal protein in general? Should we farm raise salmon? Some of those questions are overarching issues that don't have anything to do with genetic engineering. Does this trait impact fish health? Are there health risks from eating fast-growing salmon? This, of course, was some of the questions that were looked at in the FDA evaluation. Does this breeding method hurt the fish? Well, just to remind everyone, that transgenic founder event was made back in 1989 and has been inherited stably the same way my kids got my blue eyes through genetics. And so there's really no hurting involved there. Do they pose an environmental hazard? What's the governance? And of course, that's part of why those fish are kept in contained facilities in the highlands of Panama in a triploid infertile form. So all of that comes into it. And then the breeding method, I think, is really interesting. So does this breeding method pose new risks? Can this phenotype be achieved using other breeding methods? Absolutely. You know, animal breeders, that's all we do really, is breed animals that grow more efficiently, typically using better feed conversion ratios. That's what traditional breeding does routinely. And so that is something that I think is commonly seen. If you think of today's broiler population versus the 1950 broiler population, they grow incredibly rapidly relative to that. So breeders do that quite routinely. So what's the unique ethical issues associated with this particular application? And finally, what about Scott's hornless polled Holstein that was discussed yesterday? So here we've got a cow that's been gene edited for agricultural purposes to be polled. So what are the unique ethical issues here? Well, there's the overarching ones. Should we eat beef or drink milk or eat eggs? Does hornless impact the health of cattle? Well, I might argue in a good way because they're not having to be dehorned and they're not hurting each other. Are there health risks from consuming polled cattle? Well, if you've ever had certified Angus beef, you will know that there are no health risks associated with consuming polled cattle. Does this breeding method hurt the cattle? Is it a food species? Do they pose an environmental hazard? Generally, probably I would argue cattle do not. They're pretty much in a pen. I guess you could let them out. And the governance, I think, is a little unclear right now in terms of whether that falls under regulation or not, because this particular phenotype is seen routinely in other breeds of cattle. This is a naturally occurring allele that's present now in Holsteins. Does it pose new risks? I'm having a hard time coming up with that. Maybe off-target effects. Has it already been achieved? It could be if you integressed it from Angus. You know, if you did that, you're going to have issues with regard to bringing along all the Angus genetics you didn't want or the beef genetics you didn't want. If it comes to where it's very difficult to get an animal like this approved, then you may only have one animal approved, and that comes along with a whole bunch of issues associated with inbreeding and having founder line effects. You'd like to be able to have a large number of animals that have this edit so that you don't reduce your genetic diversity. As an animal breeder, I count on that variation to make genetic improvement, and I don't want to have a single founder in that particular situation. So let's move on to the topic of the conference, which is research animals. And I just want to use the example of mice and what we currently do in terms of breeding methods, right? So mutagenesis, gene knockouts, genetic engineering, how we currently make mice that are used in research. And of course, they're by far and away, as was discussed yesterday, the largest use of genetically engineered animals. And so, you know, the overall arching question, should we perform research on animals? All of that stays irrespective of the breeding method we used it. Does the breeding method impact health? Absolutely, if we're targeting animals to potentially mimic human disease. Are there non-animal substitutes to reduce all of the typical things that come into experimentation with animals? You know, does the breeding method hurt the mice? Is this a food species? No. You know, at the current time, they have enforcement discretion exercised despite the fact that they are a genetically engineered animal. And can this phenotype be achieved with other breeding methods? Well, a lot of what we're doing with gene editing, you could do with E&U mutagenesis. It just takes you a really long time to get there. And so, you know, what are the unique risks here? And I spoke to my colleague Kent Lloyd to get a feel for what sort of numbers are we talking about here. And he estimated there's about 50,000 live genetically engineered mouse lines in the United States. And that's based on kind of your estimate of how many genes there are and the different alleles that might be present. And if you consider all the lines that are frozen and stored as germplasm, there's likely more than 500,000 different genetically engineered mouse lines that have been developed before gene editing comes into play. And so a lot of the ethical concerns associated with genetically engineered animals surely applies to all of those as well before we start specifically getting into gene editing. And of course, there are issues associated with ENU lines, for example. There's thousands of off-target effects. If you got an edit in the gene you wanted or a mutation in the gene you wanted, you probably got one in every other gene as well. And according to one estimate, we're using 10 to 50 million genetically engineered animals for research at the current time. So back to the question of this conference, and that is, what's unique about gene editing animals and research? Because that's really, I think, the questions that I want to tease out. And I sat and thought hard about what are the unique issues associated here. And so one of them was the gene editing by research, so the use of gene editing for research, and that is there's some beneficial implications. One is the possibility of reducing the number of research animals used due to the efficiency of gene editing as compared to genetic engineering or ENU mutagenesis. There's less off-target effects than random ENU mutagenesis, which is good for disease models and also requires, it's more reproducible. You can specifically generate the exact mutation you want to replicate potentially a human disease rather than hoping that you might get it, which is what we currently do. And you can perform targeted gene knockout in species that are more useful as models of human disease as compared to mice, as was discussed yesterday. One concern is the inexpensive nature of the technology opens up the use to many more laboratories. It democratizes the technology, as was said yesterday, which potentially uses many more research animals, but potentially you're doing much more research. And so you might argue, well, you're doing more research, but the research animals you're creating are much more suited to their purpose, and you're actually generating a lot more data, which presumably is the reason you're doing the research in the first place. And so somewhere there has to be an evaluation of, well, okay, there might be more animals getting used, but you're getting a lot more data out of that. So what does that equation look like? And then finally, that intersect. That's what I was looking for. What is it about the intersect of all three that is unique in this space? And I think there are a couple of things that are unique in this space. One, I think, was discussed yesterday, and that is you can perform targeted gene knockouts in species that this was previously an unobtainable technology for because we didn't have embryonic stem cell lines like we did with mice. And that basically opens up the possibility of targeted gene knockouts in much higher order species all the way through to non-human primates. And that's got some ethical concerns associated with it. So I think that's one unique thing that's associated with this breeding method and research animals. You may have environmental risks associated with inadequate containment of research animals from species that have the potential to impact wild populations, and I think the gene drive with insects there is an example where you could envision the need for for very tight containment. And I guess the final one to me is, I don't know if it's an ethical concern. It is a little bit of an ethical concern, and that is, to me, it's a little bit ambiguous, the governance and regulation of this technology for research purposes, because it's kind of triggered by this ill-defined process of genetic engineering. And I think even in this room, we're having a hard time getting our head around what genetic engineering is. And it's because genetic engineering kind of has a formal definition, but genetic modification doesn't. It's an ambiguous, amorphous thing. And when the public hears genetically modified organisms, they think an ENU mutagenesis modified organism is the same as a gene edited, is the same as a genetically engineered. And yet our regulatory is kind of triggered by this genetic engineering process. And it sometimes is triggered depending upon what the purpose of the edit is. And so I think we heard yesterday, if we've got animal or insects edited, like the moth that was going to be discussed later for agriculture, then the regulations go through the USDA. If it's an insect that's modified for disease purposes, then the regulation goes through the FDA. And then just looking at some of the global discussions around this particular topic of gene editing, there's some discussion that if the gene edit is anything less than 20 base pairs, it wouldn't be subject to regulation, but if it's 20 base pairs or more, It would. Where did that come from? I mean, that as a scientist, I just don't understand the rationale for that. That does that arbitrary, to me, arbitrary number. There are some justifications for it. Makes it really difficult to know exactly, you know, so if I make a genetically engineered pig for research, it's going to go under a certain regulation, but if I make a genome edited rat with the same mutation, then it wouldn't be subject to the same regulations. And so this kind of ambiguity in terms of the whole regulation around this, I think, is a unique ethical issue with this particular intersect, because it's really a little bit murky at the moment, in my opinion. So in conclusion, I think it's important to contextualize the ethical concerns associated with the genetically modified animal application on a case-by-case basis, as was discussed earlier, because there is an importance in terms of what species you're working with, how you made the edit, and what the purpose of the modification for, because all of that affects the ethical concerns associated with it. And of particular interest to me is to avoid conflating long-standing ethical issues and lumping that all on this one gene, method to make genetic modifications. There are many ethical issues associated with the use of animals in general, the use of animals for research purposes, animal welfare, and the intentional development and use of genetically modified animals for research purposes. And I think the ethical concerns that are uniquely associated to gene editing to modify germline animals is a subset of that greater set of concerns, and that really is the nugget that is really unique to this particular breeding method. So with that, thank you very much for inviting me.